Today we will do regulation of gene expression. We will do this topic in a very easy way so that you can understand it very well. So let's start. Regulation of gene expression that is done at different levels. Like one is first one transcription level. Another one that is during RNA processing means processing level. Third one that is transport of mRNA outside the nucleus. Last one is the translational level. Today we will understand how is the gene is regulated in prokaryotes. As you all know, prokaryotes means living organisms which are microscopic and having prokaryotic type of cells. Like one is E. coli. Example of that is a bacteria. In E. coli, there is a one major example of the gene regulation is lac operon. Lac operon is a model for the gene regulation in which lactose, lac that identify as a lactose which act as a inducer and operon is a part of gene which has promoter, operator and the repressive region. So now you can see what is lac operon. Lac operon means it is an operon or a group of genes. There are a number of genes together function with a single promoter which have a promoter region and an code gene for the transport and metabolism of lactose in E. coli. In that E. coli, lactose will be metabolized and it is transported inside the E. coli cells with the help of these genes. This part of the gene is called the lac operon, in which there is a promoter operator region, and one is I. I in this that indicates inhibitor, not the inducer. Inhibitor that code for a mRNA which is called repressor mRNA. Repressor mRNA that means repressor gene. So this repressor mRNA that makes a protein which is a repressor. This repressor binds to the operator region. As you can see, its diagram is written same. So this repressor binds to the operator region, and when this RNA polymerase is transcribing this DNA step, it will stop here. And further, the functional genes ZYA. These are the functional genes of this operon, which will not be transcribed. Why? When there is lactose is present, this one, inducer, this is the lactose. If lactose is present in the medium, it will act like a inducer and it will bind to this repressor mRNA and it will inactivate this repressor and it will further not bind to the operator region. So the RNA polymerase will further transcribe and form the mRNA of ZYA. That gives rise to that makes protein that is a beta lactosidase and Y that code for permease and the A that code for transacetylase. These three are all required as beta lactosidase is the primary enzyme that metabolizes the lactose which is a disaccharide that metabolize it into glucose and lactose. These both act as the energy source for the E. coli to grow. Second one is permease that increase the permeability of the E. coli, that cell wall permeability, to lactic enter lactose inside the E. coli. Third one is transacetylase, which is responsible for the acetylation process from the acetyl coenzyme A. So these three enzymes all are responsible for the lactose metabolism and it's for the entry in the E. coli so that it can metabolize and use it as an energy source. So here you can use it in the sense of inducer, there's a repressor that binds to the operator and that mode complete the process of transcription. While when the inducer means that is lactose is present, it binds to the repressor protein and makes it inactive so that it cannot bind to the operator region and further transcription and translation that go on to form our three types of enzymes which are useful in the lactose metabolism. So how this lactoperon system works means in this when the repressor is produced without any inducer, so it is a negative feedback mechanism. In the same way, there are some examples of positive feedback mechanism. So today we have learned lactoperon model, the negative feedback mechanism of the gene regulation. Thank you.